But he did a bunch of presentations at World Aquaculture, WSN, at SURF, at the Pacific Shellfish Growers Association. He was also a really good mentor. I think he directly supervised more than 5 undergraduates and maybe some more I'm forgetting about in there.
I see a few in the room right now. So anybody who was mentored by Jacob, if you could like wave for just a second. And yeah, thank you. Welcome. They were mentored.
Awards. We ran out of time to document them all, but he has lots of them. And outreach. He did a really good job at outreach, again with our partners at Elkhorn Slough and Kirsten Wasson. He did a really good job there. So you can see he's quoted in a lot of articles, and this is definitely not all of them. There's a really good, relatively new one from CBS, a little news article, so you guys should check that out as well.
Yeah, and you could see Jacob was excellent at communicating and sharing his research. I think that those interviews are a really good sign of how comfortable he was communicating his science, not just to scientists, where he did receive awards for his talks, but also to the community. And so working with the community and with volunteers was something that, I think when Jacob first reached out, he said that there were certain important elements. And actually, I want to talk for a moment about when Jacob contacted me for the first time to work. He's contacted me and said, hello, I would like to apply to Moss Landing Marine Labs and work with you and Kirsten and Luke. And Kirsten Wasson, Luke Gardner. I was new. We all hadn't worked together. And Jacob was like, this is my vision and this is my plan. And he unified us together. He drove this sort of trio with each of us bringing our collective areas of expertise. And so Jacob really shaped and designed his project into exactly what he wanted. And it's cool that he came to oysters because thinking about his path, he did his undergrad at the University of Rhode Island, where he focused on marine policy and marine science. He has had a variety of jobs. So he actually worked at Monterey Abalone Company doing aquaculture and also worked with oysters at Point Reyes. He also, looking back at his CV, he had some stints with marine mammals as well, like working with sea otters at SORAC, and also, I forgot the other mammal, vertebrates. But somehow he came back to the, he came back to the invertebrate side, where his project encompassed a lot of time spent indoors in the lab or in the aquaculture or in the aquarium room. So here he is taking care of his oysters and settling his juveniles, which you'll see a lot more of. But that also meant a lot of checking on the concentrations of the plankton and the growth rates of the juveniles daily. So he got a lot of really great microscopy practice and time. He also spent a lot of time culturing phytoplankton, which usually worked, but sometimes didn't. And so there were also, like, Jacob is a really hard worker. So that's him giving a thumbs up even inside of that as he's repairing that carol. Jacob's also very open and a great communicator. You want to say anything? Okay. So also, he really was in his element out in the field as well. And so hopefully many of you were able to go out and join him during his field experiences. This is him smiling with an oyster, measuring them and looking very clean out in the field. And that's sometimes the case. But, he worked hard out there. And you can see him and volunteers. And also there's Kirsten, also co-advisor out there putting out tiles of oysters in the slough. It started off with putting a lot of the baby oysters from the hatchery out into their respective outplanting containers, but quickly went out into the mud. Sometimes that required some really creative ways of slogging through to get to where he needed to be. But always through all of that, he was smiling, and it was just such a pleasure and a joy to work as part of Jacob's team. And I hope we can all give him a hand as we hear about his thesis research today.
Thank you, guys. Wow, okay, thank you. That was really, really nice. And I already broke my fidget toy, so you guys be nice to me too. And honestly, I'm they mentioned the team that we built, and I'm looking around this room, and every single person in this room has been on that team. And I'll get to acknowledgments later, but seriously, every face I see has touched this project in some way. And I really hope to emphasize the team effort on this presentation. So, well, let's get it out of the way. Who's eaten an oyster? Good. Okay, good. If you didn't raise your hand, try it soon. It'll be worth it. Let's get started. So, first off, my research at the Elkhorn Slough and Moss Landing Marine Labs is located on the unceded land of the Juaneron and Guacharon peoples. This area was home to a settlement called Loquiosta in the region of Kalanduruk, or Ocean Home Place. Elkhorn Slough is within the traditional territory of several indigenous groups, including Awaswa-speaking peoples taken to San Juan Bautista Mission and Mission San Carlos, represented today by the Amamutsan tribal band. It's worth taking a minute to acknowledge that the oysters we're studying now were part of a long-standing landscape management system that we can learn from today to understand restoration. Ecological knowledge is at the heart of the relationship between people and place. Restoration is an opportunity to evaluate and shape that relationship for future generations. Indigenous perspectives and the many ongoing Native stewardship organizations will be leaders in developing effective, long-lasting restoration programs. So there are few places where the relationship between people and place is more visible than estuaries. Estuaries are where the rivers and their entire watersheds meet the sea. Estuary ecosystems include bays and harbors, and the ecological knowledge around these places has been an important part of human history as long as people have been living and traveling around our coastline. Maybe you yourself have been experienced an estuary or wetland in your recent time or around here. Let's see, maybe show of hands again. Who's been to an estuary? Who loves an estuary? All right, and if you didn't raise your hand, I encourage you to just look behind you for a second, out that window. Okay, good, now you've all been to an estuary. So that's awesome. And as I said, we're currently here in Moss Landing, where the Elkhorn Slough was once the river mouth of the Salinas River. So this estuary right behind us collected all of the water from the Salinas Valley before it entered the Monterey Bay. Okay, we'll just do one more. Who lives in an estuary or an estuary region? Another show of hands. Okay, that's pretty close, but you guys who raised your hands are like 40% of the United States of Americans who live in estuary landscapes. So in estuaries, many habitat types work together to comprise the ecosystem. We have salt marshes, seagrasses, mudflats, and even sand dunes, among several others. They work together to protect the land from storms. They protect coastal near-shore habitats from the landward runoff. They provide migration stops for birds, marine, and terrestrial species. They're also nurseries for many baby fish and crabs and important prey species. Overall, they facilitate the nutrient and energy flow between land and sea. It's a pretty big deal going on right in our backyard. So for these reasons, estuaries are home to astonishing biodiversity, not to mention our largest cities. Diversity in the estuary includes an enormous variety of human uses, along with the biological community. Here, just in this picture of Elkhorn Slough, you can see agriculture, you can see cattle, dairy, and ranching. You have a power plant. What else? We got 2 marine research stations that are pretty good. We have fishing, commercial fishing. We have shipping, maritime shipping, and a huge thriving recreation industry that I hope you have all participated in as well. So with all these processes and habitats and activities going around all year long, with simultaneous freshwater and saltwater inputs, you can imagine there's a lot of change happening very frequently. Estuaries change with the seasons, with the weather, and with the tides. So how do these ecosystems that go through so much change so frequently support such high biodiversity all year long? One answer to this paradox is a particular group of organisms called foundation species. Foundation species build habitat and modify the environment in ways that strengthen those ecosystem functions. In estuaries, foundation species include mangroves, corals, a little more tropical, marsh grasses and seagrasses, featured prominently here in Elkhorn Slough, and of course what you're all here for, the oysters. Healthy populations of these species have ecosystem-wide impacts and determine regional community composition. Specifically, they do this through really important non-trophic interactions, meaning they facilitate mutualistic relationships with other organisms not based on eating or being eaten. Although that happens as well, as you guys demonstrated. There are two main types of non-trophic interactions that make foundation species so influential. Foundation species build habitat for other plants and animals to live on, and their environmental modifications make the habitat more suitable for additional organisms. Think of a tree. Lots of animals live on or in trees. The trees provide oxygen for a cleaner atmosphere. Even the tree roots modify the soil in ways that other plants and animals depend on. I particularly enjoy the tree's shade and the water they collect on their leaves. So this graphic was produced by Aaron Ellison and his team at Harvard to show how the loss of foundation species, in this case the eastern hemlock, reduces the number of associated species, but also the connections between those species. Now I understand trees in New England might be a little foreign, but how about a more local example? Kelp is an iconic foundation species here in California, and it's currently undergoing a dramatic decline, causing the entire habitat to shift to an entirely new urchin-dominated community. Of course, some foundation species do also provide nutritious, delicious, trophic interactions. However, for this exact reason, oysters have suffered severe declines globally. Consequently, their ecosystem functions also remain depleted, and restoring oyster populations supports the recovery of not just an important food system, but it strengthens the entire community. Oyster restoration is now an important and expansive aspect of conservation around the country. In the Chesapeake Bay, pictured here, and Long Island Sound, two of our largest estuaries, the loss of oyster reefs have made the coastal communities much more vulnerable to impacts from climate change. Large-scale oyster restorations is now prioritized in these regions, and they have dramatically benefited both the environment and the people there. So we hope to take this model of oyster restoration and apply it to our native West Coast oyster. The Olympia oyster is our only native oyster species between British Columbia and Baja California. Historically, they were harvested in abundance throughout this range, and records from Puget Sound, San Francisco Bay, San Diego Bay, and our Elkhorn Slough show how instrumental they were in the development and commercialization of our estuaries and our harbors. So let's talk about this wonderful, amazing animal. Just by growing, they build habitat. You can see really, really hard shells provide substrate for animals to live on, and algae, of course. Oysters grow together in these complex clusters, and giving even more space for animals to live in between and hide in those corners and crevices. Aggregations of these oyster clusters go on to prevent shoreline erosion by stabilizing the sediment beneath them. Oysters modify the environment as well, just by eating. They filter feed by pumping water through their gills and collecting everything that passes through. Food particles are eaten and deposited as poop. Non-food particles are sorted and collected into little balls of mucus and spit out onto the seafloor. So these deposits provide nutrients and a food source to animals and plants on the seafloor that wouldn't otherwise be able to obtain those nutrients or energy. Furthermore, the cleaner water that results from the filter feeding allows more light to penetrate the water, and therefore more animals and algae can grow, again increasing further biodiversity from the organisms associated with the vegetation. Shell building also removes carbon from the water and stores it in their calcium carbonate shells. Of course, I've mentioned and am very enthusiastic about the third main way oysters support communities. They're a widely popular protein source for many predators. So that's a lot of really important ecosystem functions for such a small animal that pretty much looks like a rock. Wouldn't it be so cool to study all those impacts and how it affects our community here in Elkhorn Slough and Moss Landing and our beautiful lives? Well, young Jacob, there are not enough oysters. The Elkhorn Slough has a really similar story of oyster depletion following harvesting and a long series of land use changes, and there really aren't currently enough to affect the community. You can see in this picture pretty much what a lot of the slough looks like, a rock that used to have a bunch of oysters on it, and then additionally covered in mud. So they're having a really hard time with this muddy, sedimenty habitat, as some of those pictures showed also. I mentioned how the Salinas River used to run through the Elkhorn Slough. Well, that whole river was diverted when they built the harbor in the 50s. That cut off the main freshwater input to this estuary, along with for agricultural diking, draining, flooding, and nutrification across many sections of the slough. So really, the oyster's habitat today is fundamentally different than it was 100 years ago when they were presumably thriving. Additionally, it's an isolated population. We're really far from the nearest Olympia oyster populations. San Francisco Bay, 100 miles to the north, and Santa Barbara, over 200 miles to the south. So there's no input of new larvae going into the slough from these other populations either. Today, the remnant population of oysters is scattered across in really low densities. And this makes reproduction almost impossible because the oysters can't move and must rely on a mate nearby to fertilize their eggs. The combination of environmental changes, no outside inputs, and the low population density has led to regular recruitment failures in the Elkhorn Slough, going back to when monitoring began. 2007 was a good recruitment year with new oysters appearing, but since then, only 2012 has been the last recruitment in the slough for new oysters. So this graph shows the recruitment density on ceramic tiles in 4 sites around the slough. And as you can see, that's 2 years of significant recruitment in the last 15. As those oysters that were born in 2012 grow older, they die off, and the population density becomes even lower. By the time this project began in 2018, fewer than 1,000 oysters were living in the entire slough. So at that point, the reserve partnered with Moss Landing Marine Labs to begin conservation aquaculture for oyster recovery. Conservation aquaculture is specifically effective for isolated, low-density populations that are consistently failing in reproduction and recruitment. Because today there is sufficient habitat for the oysters, there's hard substrate available from rocks and from other man-made structures. We didn't prioritize that aspect of restoration and chose to focus on the isolated population and the low density factors through conservation aquaculture. What we're doing is providing a new input of baby oysters into the estuary by facilitating hatchery reproduction. So we're covering the input of new larvae in our hatchery here at the lab. It also improves population abundance because we bring those now baby oysters into the slough, and we can strategically transfer them to sites in a process known as outplanting. So we increase the density at target sites through this outplanting effort, and hopefully that will instigate natural reproduction as the outplanted oysters grow and mature and are close to each other. So on the left, you can see our oyster baby tanks in the hatchery here, kind of down the hallway. And then on the right is one of our outplanting sites on a mudflat in Elkhorn Slough. The problem is we can put as many oysters out as we like, but the ultimate metric of success in oyster restoration is natural reproduction and recruitment, which is currently still not occurring. We don't know what that initial underlying cause of recruitment failure was before monitoring began. But for this species, there are 4 reproduction stages that comprise the recruitment process, and we're going to look closely into that. So first, sperm is released by males, and the nearby females collect the sperm and fertilize their eggs internally within the mantle cavity. So that is the density-dependent aspect of reproduction, because they have to be close by to each other. Females brood embryos and develop the larvae inside their mantle for 2 weeks before the larvae are released into the water. They'll live as swimming, free-floating plankton for another 2 weeks before they settle and attach themselves to the most favorable, delicious-looking hard substrate they can find. So the oysters will then spend the rest of their lives in that spot. So they actually can be pretty choosy about it. This is cool. I have this video of oyster settlement. Here in the lab, you can see the oyster with its 2 shells and a little foot sticking out of its mouth. And it uses that foot to crawl around the substrate, kind of tasting the biochemistry, the biofilms, and whatever else it is attracted to. I really don't know what they like, but it'll use that foot to find it and then eventually attach itself. So that's pretty cool. So it turns out it seems straightforward, but this process gets a little more complicated when we actually try to make it happen in the lab. But we'll talk more specifically about those challenges later on in the physiology section of this talk. If any of these reproduction stages fail, then all of recruitment fails. We identified 2 life stages that we think have the most potential to cause reproduction failure here in the Elkhorn Slough. First is the juveniles in their first year after settlement, and the other is the brooding adults. These life stages may be restricting reproduction, the reproduction cycle, and therefore creating bottlenecks in the population growth. So juveniles have higher risk of mortality than adults. They're small and thin shells in this stage. You can see in this picture, looks like half of those oysters are dead, where you have a oyster body, that black dot, in these see-through shells, super thin. Not very good at protecting from predators. They're vulnerable to parasites and stress from environmental conditions as well at this stage, such as desiccation, freshwater inundation, or temperature fluctuation. The second stage, the brooding adults, you can see in this picture holding their larvae in the gill structures. That's an oyster mouth, and it's kind of open, larvae living in the gills. Larvae in your gills can probably restrict your food intake pretty significantly. They are literally in the direct pathway of the food and probably also disrupting the pumping mechanism that they need to actually draw the water in. The parental care for keeping larvae and clean and the parental care for keeping the larvae clean and oxygenated may also be a significant energy cost as well for the oysters. We don't think of the oysters as being super great caring mothers, but they do put a surprising amount of effort in, and we put effort into that in the lab as well. So these effects together we call sublethal. They're not going to kill the oyster outright, but they will negatively affect its energy balance, and reduced energy stores can limit the reproductive output for the current and future broods. So these two life stages are going to encompass the main objectives for this study. First, our goal is to improve conservation aquaculture for this species, which is generally underrepresented in commercial aquaculture. So not a lot of people are growing these in hatcheries or in any kind of lab setting. There are a few in Morro Bay, Puget Sound, and UC Davis at Bodega Bay are growing these species, but it's very experimental at this stage. Results from these experiments are important to scaling up the aquaculture production and expanding their distribution in the estuary. For the second part of this study, our aim was to leverage aquaculture techniques to study the oyster's ecology. We tested the oyster's physiology to understand changes in their feed intake and their excretion deposited during the reproduction period. So essentially for this project, we're looking at restoration through two perspectives. First is, what can we do to enhance the juvenile success after we put them in the field? The second is investigating the underlying causes that may be preventing recruitment in the future. We designed physiology experiments to investigate the hypothesis that brooding has a higher energy demand than other life stages. Through the rest of this presentation, you will see one of these two icons, iconic oysters, little babies and big fat adults. And those icons will orient you to which objective we're working on in the slides. So this two-lens approach really is only possible because we're operating in the aquaculture setting. So I'm going to describe to you 2 years of aquaculture designed to improve restoration and better hatchery efficiency along with knowledge of this species. And that's like everything I know, so sit tight. So the first thing they told me when I arrived here, not only are there not enough oysters, but if you really want to study them, you're going to have to make them yourself. So after that pilot study in 2018, where we're like, yeah, we can do this, we set up our hatchery here at Moss Landing Marine Lab. This was all possible because of this insane, incredible aquaculture team we have here at Moss Landing. Andrew, Kim, and Ava, Salmi especially, have just made that room an oyster wonderland, and I love being in there. If you couldn't tell from Amanda's pictures, I love being in there, and we have a lot of fun. So conservation aquaculture isn't entirely new. It's common for population enhancement in freshwater habitats. So you can think of stocking fish in a lake or the salmon hatcheries in the Pacific Northwest. All population enhancement, but not necessarily for restoration of a depleted or threatened species. Recently, this practice is being adapted for foundation species. That includes coral, mangroves, some marsh grass, along with the oysters. Olympia oysters, as I mentioned, are new to conservation aquaculture, but it's emerging as a really important strategy for restoration throughout the range here in the West Coast. We're modifying commercial aquaculture techniques from other types of oysters to adapt and grow our native species. So to start, we collected adult oysters from the slough in 2021. Turn the lights down low, temperature up, maybe some soft music, and larvae spawn. They settle onto substrates that we hang in the tanks. So these are clamshells that we actually collected directly from the slough, and you can see dark little spots are all baby oysters attached. So they grew here for a few months before we brought them out into the slough, outplanted in the estuary where they actually can grow pretty quickly, and we've seen are also able to reproduce in just 1 year. So the first year, 2021, we outplanted 10,000 oysters into 4 main experiments at several sites around the estuary. We had 3 central questions about our methods. When to outplant, or how long should we keep these oysters in our tanks? Where to outplant, including both tidal height and sites around the estuary, and how to outplant. Will our outplanting design itself affect oyster performance? For these experiments, we designed modular units made by hanging substrates, those clamshells you saw in the tanks, from vertical PVC stakes. Oh, look, it's our same little friends. So these guys are hanging on the tanks, and we'll watch their growth and survival in different treatments. These restoration stakes are readily adapted for various treatments, for easy deployment, and for replication. Specifically, we tested growth and survival based on outplanting size and age, tidal elevation, substrate type, and caging for predator exclusion. To test the effect of outplanting size on survival, we studied oysters from 3 different ages outplanted at the same time. So in 2021, our oysters produced 3 cohorts of larvae. Each of these cohorts spawned about 7 weeks apart. So cohort 1 spawned in July, August, and September for cohort 3. So they were all different ages. The longer they grew in the hatchery, the larger they were when they were outplanted. These bars represent their average size when they were outplanted for a range of about 3 millimeters in length to 18. So as you can see, each cohort was significantly larger than the next when they were all outplanted in December. These brackets up here show that statistical difference from NOVA's comparing the size. As you can, so I kind of mentioned it before, but size increases shell thickness, stress tolerance, and protection from predators. At the same time, delaying outplanting, keeping them in the hatchery, increases our cost and labor in the lab. And it can decrease the oyster's tolerance to change if the oysters actually become acclimated to the aquarium conditions itself, which are really stable, and they wouldn't be used to the dramatic tidal, weather, seasonal changes that I described before. So as we expected, the largest oysters had the highest survival, and the smallest had much lower survival. There was actually no significant difference between the two, between the middle size and the largest size when it came to 1-year survival. This graph shows their survival between December and July, when we would hope that they start reproducing. So was this difference due to size, or maybe was there a cohort effect that benefited oysters just because they were born first? To address this conflict, we also ran an additional experiment with this smallest cohort 3. We split cohort 3 into 3 groups and delayed outplanting for each group by 6 weeks. So the x-axis here shows how old they were when they were outplanted. They all spawned in September, but were outplanted. We waited a while to outplant them, so they were all significantly different sizes. Again, the bars show the average size, and the brackets the significant difference between each size. But I want you to notice the range of sizes is actually much smaller. So outplanted in December, again, 3 millimeters in length, but the last outplants in February were still just around 10 millimeters in length. Whereas cohort 1 and 2 from the last experiment were up at 12 and 18 millimeters in length. And I think in the oyster world that makes a big difference. In this case, same age oysters outplanted at different times. The oysters had equal survival in the slough immediately following outplanting. So this is in contrast to our cohort comparison. You can see all three groups started at different sizes, but each averaged just over 50% survival in that first period after outplanting. My hypothesis for this difference between the two results is that the advantage that comes with size may only occur above 10 millimeters. And these smallest size groups would have been below that threshold and therefore all equally vulnerable to the stressors of the slough. So survival improved when our oysters were outplanted larger than 10 millimeters. And for us, that was between 12 and 14 weeks of time in the hatchery. But there was no difference between sizes when they were outplanted smaller than 10 millimeters. No difference in survival. So now we know when to outplant. The question is where to put them. Olympia oysters are mainly intertidal. Tidal elevation affects thermal stress, food availability. They can only eat when they're underwater. Predation pressure. Some predators come from the sea. Some predators come from land. And both at low tide and high tide, maybe they have different predators. In the intertidal, there's also a lot of competition for space. Lower tidal elevations, and you can see this is the lower tidal elevation, and this is the higher tidal elevation. There's oysters on each of these tile substrates. At the lower tidal elevation, there's a lot more competition from other organisms that also want to attach themselves to that tile. So the opportunity and the space for growth can be really restricted lower down, even though they are getting more water, more oxygen, more food. There's a balance going on here, and we're going to check it out. So we found that the lower elevation had higher survival initially, but it actually wasn't different after 1 year. So the asterisk on this chart shows when the survival was significantly different, which time interval, and then in the end, again, about 50%, a little lower for survival. We observed this pattern at all three sites that we tested and found no difference between sites. It seems like the tidal elevation may affect the earlier stages differently than it does after the oysters are 1 year old. There may still be tidal elevation effects in the larger oysters, but it wasn't in growth and survival. So all the oysters in this tidal elevation experiment were the same size when they were outplanted, and the lower elevation was advantageous for outplanting younger oysters, younger and smaller. So we also wanted to assess the success of our outplanting design. Substrate selection is important for restoration outcomes such as settlement preference, naturalistic clustering, and accessibility for monitoring. But these substrates may affect growth and survival differently. We outplanted oysters settled on clam shells that we collected from the slough, abalone shells from our local farm, and the ceramic tiles that are traditionally used for the wild recruitment surveys. We know oysters attach pretty well to each of these substrates, but when we tracked their survival in the slough, again from December to July, we found that the oysters on the abalone shells had the lowest survival. We think this was most likely due to also having the highest settlement rates. So the abalone shells, when we put them out, had the most oysters on them, and therefore the most competition. So the low survival really came from oysters outcompeting each other, and sometimes they actually knock each other off of the substrate. So low survival on the abalone shells. However, the two shell substrates, clams and abalone, had an additional benefit for our restoration program in that they created those three-dimensional natural clusters. That complex three-dimensional structure is really important when we're rebuilding habitat beyond just more oysters. So compared to the tiles, both shells you can see in this picture, actually, there's an abalone shell and a clam shell in this picture. They're just covered in oysters, and that's a pretty significantly large cluster, and we do find a lot of animals living in there every time we go and check them. So lower survival on the abalone shells, but not the flat two-dimensional result that we got on the tiles. So far, oysters' survival increased when they were outplanted above 10 millimeters at mean lower low water and on clamshells and tiles where the competition was minimal. I don't know why the oysters preferred to settle on the abalone shells in such high densities. There's a lot going on with the abalone shells. There's a lot more texture, but really the cover on the abalone shells was just so much more that result wasn't too surprising given the way we started. So cages are standard in aquaculture for single unattached oysters, but less common for these large oyster clusters. Cages are important for restoration because Elkhorn Slough has voracious sea otters. Predator exclusion may protect vulnerable juveniles, but can also affect the water flow and conditions that are important for growth and survival. Oysters clustered on clamshells and abalone shells were compared inside and outside cages. And you can see some oysters in that little homemade makeshift cage. For the experiment, we used these commercial SIBA baskets, and you can see how clean and healthy those oyster clusters are in there. Cages did have a positive impact on survival. However, this was not likely due to predation from sea otters, but rather the oysters in the cages that got detached from their substrates were retained in the cages, as opposed to oysters that got detached outside of cages, which would fall straight into the mudflat and quickly be buried. So cages worked out. Overall, our oysters in the slough did better when they were outplanted after reaching 10 millimeters at mean lower low water compared to the higher elevation on less dense clamshells and tiles and inside the cages, mainly to keep them out of the mud. And that's a pretty special Elkhorn Slough problem. Surprisingly, growth was about equal, and oysters in all of these treatments averaged about 50 millimeters after just 1 year. It's pretty big for Olympia oysters in California. So from these studies, we also found new sites to expand our restoration into that previously had no existing oyster population. So that's a really, really good sign that we can inhabit sites that don't necessarily have oysters now, but they'll still do well. These 2021 experiments directly informed our restoration approach and site selection for the subsequent outplanting in 2022 and 23. Now that we have a better handle of our aquaculture methods, we can plan our restoration to mitigate vulnerabilities during juvenile growth, such as high elevation and competition that caused the detachments in the first place. Stake designs, the stake design, clamshells, and cages fit the Elkhorn Slough's restoration priorities for maintaining high densities, for long-term monitoring, and for adaptive management. We successfully prevented burial, encouraged naturalistic clustering, and avoided predators for now. Outplanting was a really great way to engage with the community. Every time we brought oysters to the slough, it was a whole team, different groups, volunteers, public events, and it was a really, really special opportunity to engage the local community with their ecology and natural resource management. So for objective 1, we observed that oysters grow quickly to large sizes in many sites around the slough. However, we still had no new recruitment in the slough after putting those 10,000 out. So the question remains, how can the oysters have high growth and survival, but at the same time, no viable reproduction? Well, if an oyster's energy demand increased during reproduction, such as the brooding period, then they may have enough energy to grow and survive, but not enough to successfully reproduce. In other words, they're getting enough food to live on, but not enough to produce offspring and really live the lives they want to. So for objective 2, I studied the oyster's energy balance during brooding and non-brooding periods to determine if the reproductive life stages are vulnerable to these sublethal effects. Ultimately, restoration success relies on consistent natural reproduction. To study the energy balance of an oyster, we can think of how the energy is used. First, oysters filter feed, and first the oysters filter feed. They get all their energy from the food they eat, and we call that consumption. There are no other energy inputs into the oyster. Some of that food is rejected as pseudofeces, and that energy never reaches the animal. After that, some energy is consumed and used for metabolism and the basic life requirements, the requirements of living. We measure this as respiration. So we measure those basic requirements of life through respiration. Some energy is not usable and gets excreted through urine and feces. And the energy, and all of that energy becomes available to other organisms. So finally, all the energy left after these processes can go toward production, which describes body and shell growth as well as reproduction. So this equation on the left describes how the animal's energy is balanced between those processes. Again, all of the energy they can possibly use comes from what they eat. In order to understand the oyster's energy budget, we can rewrite the equation to determine how much energy is left for production after those processes. The amount of energy allocated for reproduction can be determined by subtracting each type of energy use from the total amount of consumption. And what we're left with is the amount of energy available for growth and reproduction. Specifically, we designed experiments to assess the relationship between feeding and the excretion deposits, pseudofeces, and feces. We compared oysters' feeding and excretion between brooding and non-brooding periods. The brooding period, as we saw that picture earlier, is marked by larvae occupying the oyster's gill structures. This diagram shows the pallial cavity of a Chilean oyster with brooded larvae inside. You can see these white circles represent the larvae in that gill cavity there. So Mardones Toledo et al. Showed that the Chilean oyster actively circulates the larvae, you can see the arrows, between the different feeding grooves for that aeration and keeping the larvae clean. But that is, again, directly in the path of the oyster's feeding. So imagine you have your babies in your mouth and then you also try to eat through a straw. That's what they're doing. It's not easy. It's not hard to think of how that could be disruptive to their energy intake. Not only is this activity blocking the food intake, but it likely costs extra energy for those movements themselves. So to investigate the energy balance between the two stages, our experiments required two concurrent aspects: tracking the reproductive timeline over the entire hatchery season and individual feeding excretion trials to examine the relationship between the feed intake and the energy deposited during that reproductive period. So during our first year, when we were getting all those juveniles ready for their field experiments, we noticed a difference in the feeding rates in our adult oysters while they were spawning. We took this opportunity to test our hypothesis about the restricted feeding in 2021 with groups of oysters living together in high-density tanks. We called those adult oysters broodstock. So we measured the clearance rate of those groups of oysters to monitor their feeding. So clearance rate is a calculation of how much water is passed through the oyster in a given amount of time. In this case, we measured for 1 hour. This video is showing one single oyster, actually in New York, in a 1 liter container. The 2 containers started with equal amounts of food, and as the oyster eats, you can see the clearance rate, quite literally. So for this experiment, we had 20-liter tanks with 80 adult oysters in each tank. They were fed 300,000 cells per milliliter of live microalgae Tetraselmis. We sampled the decreasing algae density in the tank over 1 hour to measure the clearance rate of the entire group. We measured the clearance rate before and after males spawned in each tank. So this is pretty interesting. We did find differences in clearance rate. We measured each tank twice, once before the males spawned, so before fertilization happened, and then once after the males spawned while brooding was happening. We were able to confirm this by tracking the reproduction and seeing when the larvae were reproducing in each tank. So we were able to confirm each tank had brooding oysters in it while we were measuring. And a big shout out, much of this data was collected by our undergraduate Saheli back then in 2021. So it's interesting that they would change their feeding rates on that group level. If they're eating less during brooding, we could actually be harming the oysters by giving them too much food. It's hard to imagine, too much food, I know, but that would actually could cause them to waste energy if they're forced to sort more food and produce more pseudofeces. That's an energetically probably pretty exhausting. So we might be worried that if they have too high of a feed density, they're actually spending more energy than they would with less food. So seeing this result, and it's, again, very visible after brooding, or after that male spawn and before. So seeing this, we decided to track their feeding through the entire breeding season the next year, 2022. Tracking their feeding will help us understand how their physiology and energetic needs might change during this critical phase. So how do you track oyster reproduction? We saw the 4 stages, but it's one thing to have a nice diagram and someone narrating for you, and it's a whole other thing to look at a tank of, not rocks, I swear, to see what's going on inside them. We can't open them up, and we can't ask them their opinions. So to compare brooders to non-brooders, we had to know when they were brooding. In 2022, instead of those 20-liter tanks, we kept our oysters in just 2 300-liter tanks. Altogether, we tracked the reproductive cycle for this entire broodstock and sampled individuals from the tank to measure their individual physiology, feeding, and excretion. From that, we were able to build a reproduction timeline based on tracking all the male spawning events or sperm releases, which corresponds to eggs being fertilized, and all the larvae spawns that occurred in the 2 tanks. This is one tank. The other tank is right over here. Fortunately, these are actually very conspicuous events in the oyster's life. We're all in there checking the tanks twice a day for those two male spawns and larvae spawns. So just to kind of illustrate how conspicuous and visible those events are, if you remember the tank from the last picture, after males release their sperm, surfactants cause a big layer of bubbles to occur. It mixes with the aeration in the tank, and you walk in the morning, and very clearly, males are spawning. I also was able to capture a video of an individual male. You see its mouth open right here, and you see a stream of not bubbles, but sperm packages that are released by the male. Those sperm packages swirl around in the current and actually from there release all the sperm cells in kind of a spiral firework looking action. So there's a few oysters. You can see them open in there, and you can see the sperm in a very, again, discrete track. And I think it's on a loop. So then larvae spawn, not as visible, but when you have a flashlight, again, very visible. It's not a snow globe, it's larvae. So all those little dots are larvae. And in this video, it's really fun when you go see them at night because you can actually see like individual larvae swimming around. The air is off and there's no current in the water. So this kind of thick layer of larvae goes all the way down through the whole tank to these oysters on the bottom. So from this record of the two reproductive events, we could retrospectively track the brooding phases between fertilization and larvae spawn in the broodstock tanks. Additionally, we found, while measuring the individual oysters, that the reproductive stages could be tracked more specifically. Individual brooding oysters eject larvae from the brood, and they are released onto the seafloor along with the pseudofeces. So you see that pseudofeces here in a pile, and these are all dead oyster larvae that had come out of the brood before being released in a spawn. So we were actually able to observe this very directly during our experiments, and this is the first time this has been documented for this species. It has been described for the Chilean oyster, again, our little cousin down there, but not specifically for this species. So this actually became a really reliable, non-invasive method for determining the reproductive status. Again, the traditional method is to open them up, which is sacrificial. Feeding and excretion measurements were taken for these individuals throughout the hatchery season, and the reproductive timeline was used to determine which measurements were taking place while the oyster was brooding and when it was not. So again, a very conspicuous observation. It's very easy to see when the larvae are being ejected from the pseudofeces. And you can tell they're dead because they're totally see-through, but not all larvae that get ejected are dead. In this video on the right, you can see two live larvae and one dead one and one undetermined one that are actually just stuck in the pseudofeces. They're not swimming, they're stuck on the bottom, and they're not going anywhere, although very much active. Cool. So now we're recording all of the oysters' reproductive activity. Let's start measuring their feeding and their excretion. We tagged 8 individuals from that broodstock tank. We placed them in 1-liter chambers. You can see the oyster in its 1-liter chamber. It's got aeration and clean water. To measure the feeding and excretion, they were acclimated with no food in this chamber for 24 hours. They were then all given another 300,000 cells per milliliter of microalgae, in this case, the single-celled microalgae formerly known as pavlova. We can then measure the change in algae density to record the clearance rate. They were removed after that feeding period from the chamber, placed in a second 1-liter chamber with, again, no food, and all the excretion was collected from that chamber after 24 hours. So that way, we had all of the excretion from that one specific feeding event. The acclimation period, they cleared their digestive tract. They ate a known amount of food over a known amount of time. And then we collected all the excretion, the pseudofeces and the feces that came from that specific feeding trial. After the trial was completed, we could determine the brooding status by observing larvae in the pseudofeces. So these containers are what that mix of excretion looks like, the feces and pseudofeces combined from that excretion chamber. We would sort through this to find either ejected larvae or to separate the feces and pseudofeces. We could, once we sorted the feces from pseudofeces, we were able to filter it, measure the weight, the dry weight, which is the total mass, and also combust the sample in these little foil envelopes, the bane of every undergraduate researcher, are these foil envelopes. And we put those in the muffle furnace to combust it, which gives us the organic content of that excretion. So we have the total amount of excretion and the organic content of both the pseudofeces and feces, and that is really what's at the heart of the energy content in the food. So all that data was collected by Deo Scott and Natalie Frantella. That's over that summer and fall, and compiled into this really complicated looking timeline. Don't worry about the dates, but you can see the colors show how frequently these reproduction events are happening. Males are spawning very frequently, and each sperm release is not necessarily corresponded to a larvae release. So just because the males are spawning does not mean fertilization is happening. We also included a reference to the average clearance rate for those 8 individuals over the course of that 2022 hatchery season. So you can see this is the average rate. It changes and is also really highly variable over time. What was really surprising was all 8 of our randomly selected individuals actually followed the exact same trend and pattern at the same time. So they were randomly selected, but they were all either brooding or not brooding at the same time. It was a little surprising. It makes me think that maybe the tank conditions may have been favoring a female development, or there was something triggering sex switching in these tanks. So I talked about that extra level of complexity that's coming. Well, these oysters live their own reproductive lives individually and will also switch between male and female in ways that I have not figured out. It's pretty, there's no pattern that I've observed, but something is inducing these animals to switch sex, and there may be some environmental mechanism for synchronicity going on here that we haven't really pieced together in the tanks themselves. So if you're ready for another surprise, contrasting our results from the group feeding experiments, we found no difference in feeding rate between the brooding and non-brooding period. In this graph, you can see all of the trials that took place while brooding and all the trials that took place while they weren't brooding combined for this analysis. On the right, you can see all of these measurements pieced out for the week that it was measured. So you can see all 8 oysters were not brooding for this trial. They were all brooding for these, not brooding for these, and brooding for these. As variable as these averages and the variability itself is here, it averaged out to be not different. Since each oyster was given the same amount of food to eat in the same amount of time, what this also tells us is that they're not eating more or less quantity either. They're not eating faster or slower, and they're not eating more or less food. The individuals, not only were the groups different week to week, but the individuals themselves were different week to week. Some oysters did show increases and decreases between brooding and non-brooding, but this pattern wasn't even consistent between the 8 individuals. We did not expect to find this, and again, referring to the Chilean oyster, where they did see a drop in feeding during the brooding period. But then we assessed the excretion production in relation to the amount eaten. And maybe it's not that surprising that the feeding didn't change, because we actually found no relationship between the amount that was eaten and the amount of excretion that was produced. This graph shows the same trend or non-trend for the feces in orange and the pseudofeces in green. Consistently, maybe there was more feces than pseudofeces, but not in relation to the amount they ate. And this, again, was true during the brooding and the non-brooding phase. In addition to this, the feces and pseudofeces were also not related to each other. The graph looks really similar, but the amount of pseudofeces that they spit out had no relationship with the amount of food they ate and excreted through poop. So it could be higher or lower in any given trial for a single oyster. So overall, feed did not change with brooding. So the real story is in the poop and the pseudopoop. Pseudofeces production dropped during the brooding phase. In addition, the feces production was the same, but the organic material in the feces was also lower during the brooding phase compared to the non-brooding phase. So they rejected less food, and they also incorporated more of the organic material from the food that they ate while brooding compared to when they were not brooding. I think there are 2 possible explanations for this pattern. First, the oysters have higher energy demand while they're brooding. Rejecting less food and incorporating more organic material indicates that the oyster is just using a lot more energy, again, either for parental care or for maybe an increased metabolism that comes from the conditions while brooding. Another possibility is that the larvae are actively consuming a portion of algae. The larvae themselves are consuming the proportion of the algae instead of it being rejected by the adult. So less pseudofeces, maybe the adult isn't the one taking it in, but the larvae themselves are feeding. And this pattern was observed in the Chilean oyster, which we've seen have some similarities with the Olympia oyster and some differences. So we have seen this pattern of larvae eating the food themselves, especially as they get older, and that also could explain the pattern that we observed. Of course, these 2 ideas are not mutually exclusive, and it's not even unlikely that both are occurring. However, in both cases, that means the proportion of the oyster's energy intake is being allocated for larvae care, either parental care directly or the larvae themselves. Therefore, the oyster must extract more energy from its food that it doesn't take in, and this is reflected in the lower organic content in the feces. Thank you for being patient with me. Okay, so in conclusion, we tested the effect of brooding on feeding and excretion. Brooding did not change the feed intake. Brooding decreased the pseudofeces deposition, and brooding decreased the organic matter in the feces. The excretion results show a difference in the oyster's energy use. Brooding oysters took up more organic material, indicating they have an increased energy demand. While the larvae did not disrupt feed intake, they may have caused less food to be rejected in the pseudofeces. However, the brooding oyster did not compensate for this increased energy demand by eating more. Therefore, I concluded that the brooding stage is susceptible to energy limitation. The oyster will eject the larvae, as we've seen, before it stops its other energy use requirements. So it's going to get rid of its brooded larvae before it stops breathing or pooping, I guess. So oysters, as we also learned from that really complex yet beautiful timeline, have a really long reproductive season in the Elkhorn Slough. If brooding has a high energy requirement, and we've seen them in the hatchery brood multiple times in a single season, then the energy reserves are what's being taken each time. So that indicates that the feeding in the winter and the spring will be really important for avoiding reproductive failure in the summer and fall. So this project addressed our conservation methods. We found juveniles grow quickly with high survival. We developed outplanting guidelines for future conservation aquaculture efforts, and we expanded the oyster's distribution in the slough just in the last 3 years. We also were able to leverage aquaculture methods to study the oyster's ecology in our estuary. The cost of brooding may cause reproduction failure, and it will be worth investigating food availability and other conditions that affect their energy balance during this spring and the summer. We also found that brooding oysters eject larvae, which became a really nice, reliable, non-lethal method for tracking reproduction. So our goal is, so next steps are critical. Scaling up our restoration is going to take a lot of work, and specifically will require increasing hatchery production and the spatial expansion of our restoration program. We have several steps to go before optimizing our hatchery conditions, including improving the growth rates in the tanks and synchronizing the spawning so that timeline doesn't look so complicated and our efforts can be made a lot more efficient. To expand the oyster's distribution around the estuary, site parameters and feeding conditions will have to be assessed for future site expansion. I also hope that moving forward we'll be able to track natural recruitment in relation to the population density that we're actually creating. So is the density that we're improving actually leading to recruitment in the slough? Integrating physiology into the experimental aquaculture will support these goals. So I hope I've shown you the value of conducting restoration experimentally and the power of leveraging aquaculture to support ecological goals. Aquaculture is incredibly adept for experimentation and adaptive management. As we're relearning the oyster's ecology here in the Elkhorn Slough, we're shaping the restoration efforts around community-driven priorities and values defined for our estuary. This includes not just biological goals for the oysters, but also social goals for habitat uses, biodiversity, and building relationships within the community. The Elkhorn Slough has particular values for functional and aesthetic outcomes, for climate resiliency, and for accessibility, research, and education. The oysters can be part of all of those values. This year, we outplanted a total of 160,000 juveniles into 5 sites around the Elkhorn Slough. The experiments I described today directly informed our restoration approach for these new oysters. Now that we have a better handle on our aquaculture methods, we can plan our restoration around the values defined for our estuary. I have so many people to thank. It takes a brave soul to volunteer in the Elkhorn Slough. I'll start with my advisors because that was the nicest introduction I've ever heard, and I've been inspired by each of you individually. I don't even have my head on straight. We've had collaborators from Amanda's lab, from Luke's lab, from the entire Native Olympia Oyster Collective collaborative team. Lots of people at the Elkhorn Slough and volunteers, as well as the Amamutsun Land Trust Native Stewards who have been part of the project since I arrived. I mentioned our aquaculture team who were really the lifeblood for these oysters. And I have a lot of experience to thank each undergraduate researcher because I thought we had so much fun, but I learned a lot from each of you. And I really appreciated not just each of your enthusiasm, but your willingness to take on my learning style and learn right along with me. And it was really an amazing process. So really just thank you to everyone. Thank you to Dr. Kat Stacks for keeping my head on straight and my eyes in place. And with that, I would love to take any questions. Thank you.
Any questions in the crowd? Feel free to just walk up to the microphone here.
Yeah, just make a line. We don't have to look around at each other.
Okay. Great job, Jacob. Especially considering you've still been raising and outplanting oysters up until yesterday.
Yeah,
right?
yeah. We were in the slough yesterday, so
yeah, it's very
the
clear.
last of those 160,000 are now out. So.
It's very clear how much effort you put into this.
Thank you. Thank you.
I had a question just kind of going forward, looking forward with the restoration, and it's about introducing new genetics into our population, which I know can be kind of a big no-no with these isolated fragmented populations. But do you think it'd be beneficial or detrimental to introduce some brood stock from maybe San Francisco or Santa Barbara?
Yeah, that's a really good question. It's a question that our team asks each other probably every month because we do have a significant population bottleneck. I mentioned less than 1,000 oysters have directly have directly produced all those 160,000 oysters. So genetic bottleneck, 100% is a possibility. One thing that we're also focused on is local adaptation. So Olympia oysters, it's a big range from the subtropical Baja California to the subtemperate British Columbia. And we've seen that the oysters, not just their physiology, but their reproductive habits are very adapted to the local conditions. And that can be even within a single estuary. So I think there's a lot of value in maintaining that local adaptation because it's going to give our population the best chance to keep up with the changing conditions that we're dealing with. genetic diversity is hugely important as in restoration, and there's a lot of value to increasing that from other sites, especially in like a really emergent situation where you need more oysters. But I think we made it work still, and there is genetic analysis for these different generations that's going to be going on. Luke Gardner and Kevin Johnson at Sea Grant are leading that, and Anna Heasley has her project assessing some Olympia oysters from different estuaries as well. So thank you for your question. Wow, so thorough. Come on.
Great job, Jacob. This is really a tremendous effort, so this was awesome to see. But my question is, with your outplanting, no matter what substrate these oysters were on, it seemed like those pillars were kind of less conspicuous than a natural oyster settlement. Do you think that there was any enhanced predation because of these outplanting?
The stakes themselves, yeah, that's a cool question. We've seen some evidence of predation on our oysters, like a few broken shells. Nothing so dramatic at any of our sites. So I'm confident predation is happening, but it's not a function of the stake itself. It definitely could be a vehicle for a crab or a snail, and I'm waiting for the day for the otters to find them. So yeah, it's a new infrastructure to the slough, and it's going to be really worth evaluating and tracking. To speak to that, because it's not just predation we're worried about, it's also quite a bit of plastic. And we're actively testing new prototypes of substrates as well. So the shells are really nice, biogenic from the system itself. We just put out a bunch of our oysters on eucalyptus sticks, which also came from the slough. As they're getting rid of the eucalyptus, that's a very also natural substrate. So we're just balancing the naturalistic profile, the community, and the effectiveness. And the durability is really what it comes down to. 2021, we started, and we're like, okay, we're going to take a few hundred oysters. And then we found that actually the stakes that were out were falling apart. So we actually had to bring a lot more oysters in than we intended. But the restoration design itself, I'm glad you brought up, because that's something that we're always thinking about.
All right, thanks for the questions. We're a little past time, so maybe just if there's one more question from the audience or one more person. Kirstine, want to come up and ask your question?
I have a little surprise for Jacob here
Oh, no.
at the end, since I didn't join the introduction at the beginning. I would like to inform you that you've been chosen as the 2023 recipient of the 2003 Conservation Research Award, sponsored by the Elkhorn Slough National Estuarine Research Reserve and Elkhorn Slough Foundation.
Thank you.
And I have a few words. So this, and you'll get a certificate, you'll get $200 in the mail from the Elkhorn Slough Foundation. This award honors a junior researcher who's recently made a meaningful contribution in estuarine science, in particular by answering questions that inform conservation and management strategies, and various Moss Landing Marine Lab students have won this in the past. You will see Jacob's research on our webpage of past winners, but check that out, and maybe one of you in the audience will be in the future list. The reason you were chosen as this year's recipient is because of your dedication to every aspect of Olympia oyster conservation aquaculture, from being the proud oyster daddy of more than 100,000 oysters this year, along with the rest of the hatchery team, to tracking their growth and survival in the field, and synthesizing findings at conferences and in publications. But most importantly, you not only care about the oysters, but about the people who interact with them. So watching you talk to a 5-year-old participant in the summer camp for Amamuts and tribal members, or an 80-year-old hiker who comes along the trail and interrupts our restoration, it's always a delight because you are so inviting and so joyful in sharing information with them. So in summary, the Elkhorn Slough Reserve and Foundation are grateful for the thousands of oysters that you raised to help bring back slough populations, for the rigorous science you conducted on them, and for your outreach to diverse audiences. So your contributions are going to reverberate at the estuary for a long time, and we look forward to continuing to partner with you as you start your next chapter. So congratulations.
I had no idea about that.
Okay, thanks so much, everyone. The committee is going to steal away Jacob for a few minutes to just chat about some final steps. But if you have some time to stick around, we've got some refreshments, and I'm sure that Jacob would very much appreciate getting to celebrate and speak with you. And then there's also a picture back there there's a pen that you could sign on the back. Sorry?
Just sign it gently so you don't puncture it.
Sign gently. It's delicate paper on the back.
I'll know whose hole it is.
Okay, we'll be back soon. Thanks, everyone. Thank you, Jacob. Congratulations.
Should I ring the bell? Oh, maybe me too.
Cool. All right.
Yeah, so we'll just go next door.